All right, welcome everyone. We're going to dive right into one of the most incredible fundamental processes in all of biology, synaptic transmission. This is it. This is how your brain cells talk to each other. And understanding this is the key to, well, understanding pretty much everything your brain does.
So here's our game plan. We're going to follow the journey of a single neural signal, step by step. We'll start with the two main ways neurons can connect. Then we'll look at how a message is sent, how it's received, what that message actually means, is it a go or a stop signal, and finally, how the neuron adds it all up to make a decision. Okay, let's jump in.
First things first, let's look at the point of contact, the synapse itself. This is where all the magic happens, and it really comes in two main flavors.
And what you're seeing here is a fundamental trade-off the brain has to make. On one hand, you've got electrical synapses. They are pure, unadulterated speed. Think of them like a hardwired connection, perfect for things that need to happen instantly, like a reflex, or for getting huge groups of cells to fire in perfect synchrony. But on the other hand, you have chemical synapses. They're a bit slower, sure, but that delay buys you something incredibly valuable: control, complexity, and the ability to change over time. That's plasticity, the basis of all learning and memory.
So how do electrical synapses get that speed? Well, it's because they're basically a direct tunnel between two cells. These proteins, called connexons, link up to form a pore, a gap junction. Ions, electricity, just flow passively right through from one neuron to the next almost instantly. There's no middleman. It's this direct physical bridge that locks the cells together electrically.
But with chemical synapses, it's a totally different story. There's no bridge. In fact, there's a physical gap called the synaptic cleft. It's like a tiny moat separating the two neurons. And to get a message across that moat, you can't just send electricity. You have to launch a fleet of chemical messengers, which we call neurotransmitters. This little gap is the stage where all the really complex, regulated, and fascinating parts of neurotransmission play out. And that's what we're going to focus on for the rest of our time.
Okay, so let's zoom in on the sending neuron, the presynaptic terminal, and see the incredible molecular machine that launches that chemical message.
This sequence right here is absolutely critical. You've got to know this cold for the exam. First, the electrical signal, the action potential, zips down the axon and slams into the terminal. This voltage change forces open special gates that are only permeable to calcium ions. Calcium, which is in high concentration outside the cell, floods in. And that's the trigger. That influx of calcium is the signal that tells these little bubbles full of neurotransmitter, the vesicles, to fuse with the outer membrane and dump their contents into the gap. And boom, you've just converted an electrical signal into a chemical one.
Let's just pause on that for a second. Calcium is the absolute, non-negotiable trigger. It is the direct link between the electrical arrival and the chemical release. If you could somehow stop calcium from getting in, the action potential could fire all day long, and not a single molecule of neurotransmitter would be released. The message stops dead without it. So how does this happen so incredibly fast? The secret is preparation. The neuron uses these amazing proteins called snares, which act like a molecular zipper. You have some on the vesicle, some on the membrane, and they literally twist and pull each other together, docking the vesicle right up against the membrane, ready to go. They're zipped up, primed, and just waiting for the final signal. By the way, this is exactly the machinery that toxins like botulism and tetanus attack, which is why they're so powerfully paralytic.
And that final signal, the one that completes the zippering process, comes from a protein called synaptotagmin. This is your calcium sensor. When that wave of calcium rushes into the terminal, it binds directly to synaptotagmin. This binding causes a rapid change in the protein shape, which provides the final kick needed to fuse the two membranes together, creating a pore and releasing the neurotransmitters. It's an absolutely beautiful and elegant piece of molecular machinery.
Okay, the message is sent, the neurotransmitters are floating across the synaptic cleft. How does the neuron on the other side, the postsynaptic neuron, actually hear this message? It listens with highly specialized proteins called receptors.
And just like synapses, receptors come in two main flavors. On one side, you have ionotropic receptors. These are the speed demons. They are a receptor and an ion channel all rolled into one protein. The neurotransmitter binds and bam, the channel snaps open. It's direct, it's fast. On the other side, you have metabotropic receptors. These are the strategists. They aren't channels themselves. When a neurotransmitter binds, they kickstart a whole biochemical chain reaction inside the cell, often involving something called a G protein.
This is slower, yes, but it allows for much more complex, long-lasting, and modulatory effects. It can change the cell's metabolism or even wander off to the nucleus and change which genes are being expressed. So, we know a neurotransmitter binds to a receptor and an ion channel opens. But so what? Does that push the neuron closer to firing its own signal, or does it push it further away? Is it an excitatory signal or an inhibitory one? The answer lies in the simple math of ion flow. The whole thing really boils down to the flow of charged ions across the membrane, what we call the postsynaptic current. And that flow is dictated by a really simple but incredibly powerful relationship that honestly is the key to understanding all of this.
Now, don't let the equation intimidate you. It's simpler than it looks. It just says that the current, the flow of ions, depends on two things. First, G, the conductance. That's just how many channels are open. More open channels, more potential for flow. Second, and this is the important part, is the driving force. The driving force is just the difference between the neuron's current voltage, Vm, and a special voltage that each ion has, its reversal potential, or ERV. You can think of ERV as the ion's happy place, the voltage where it doesn't want to move in or out. The driving force is just the push or pull the ion feels to get to its happy place.
Now this is the single most important rule you need to take away from this. Burn this into your memory for the exam. Whatever a neurotransmitter does, its one and only goal is to grab the neuron's membrane potential and drag it kicking and screaming towards the reversal potential for the specific channels it opened. That's it. That's the whole game. So here's where it all comes together. We know a neuron fires an action potential when it reaches a threshold of about -40 millivolts. So the logic is simple. If a neurotransmitter opens channels whose reversal potential is above that threshold, it's going to pull the voltage up towards firing. That is excitation and EPSP. But if it opens channels whose reversal potential is below the threshold, it will always pull the voltage away from firing. That is inhibition and IPSP. It's just a simple comparison. And this is why glutamate is the major excitatory neurotransmitter in your brain, and GABA is the major inhibitory one. It's not magic. Glutamate receptors let in ions like sodium, and their combined reversal potential is around 0 millivolts. Well, 0 is way above -40, so it's excitatory. GABA receptors let in chloride, whose reversal potential is around -70 millivolts. That's way below -40, so it's inhibitory. It's all about the E-rev. Now, here's the thing. A single one of these little excitatory signals, an EPSP, is almost never enough to make a neuron fire. A typical neuron in your cortex is getting bombarded with thousands of inputs at any given moment, both go signals and stop signals. And it has to take all of that chaos and make one simple decision: fire or don't fire. Each little excitatory push is just a tiny blip, a whisper in a hurricane, far too small on its own to reach that critical firing threshold. So how on earth does a neuron ever make up its mind? It does what any good decision maker does. It integrates. It sums up all the evidence. And it does this in two ways. There's spatial summation, that's adding up signals that arrive at different places on the neuron at the same time. And there's temporal summation, that's adding up signals that arrive at the same place but in a rapid-fire sequence. The neuron is basically a tiny, incredibly sophisticated calculator, constantly adding up all the pluses from the EPSPs and subtracting all the minuses from the IPSPs. And the whole process comes down to this one number. At the base of the axon, a spot called the axon hillock, this grand sum is calculated. It is a constant, brutal tug-of-war between excitation trying to pull the voltage up and inhibition trying to clamp it down. If, and only if, the sum of all those inputs is enough to push the membrane to that magical threshold, the neuron fires an all-or-nothing action potential. If not, it stays quiet. And that's it. This constant, dynamic competition is the literal basis of computation in the brain. Every single thought, every calculation, every perception is the result of thousands of these little contests being won or lost. So just think about that. This entire intricate process, from calcium flooding in, to neurotransmitters crossing a gap, to this final tug of war, is happening billions upon billions of times every single second inside your head. Just so you can listen to this. Your entire conscious reality is built on this simple binary decision, repeated over and over again, to fire or not to fire. Okay, that wraps up our review. Thanks for your attention and good luck with your studies.